Let's go over this slide. So this is an injury has happened, and as a result, the injured tissue it has released certain chemicals. And what is the first step? First step is there is leukocytosis. What is leukocytosis? Leukocytosis is that the bone marrow it is now pushing more leukocytes into the blood because it knows that something has gone wrong. So leukocytes they increase in the blood, and they travel towards the site of the injury, and as they travel in the blood vessel they get closer to the wall and they get slower and slower. How do they know which is the point where they need to squeeze out? So this process is known as margination, and the endothelial cells they exhibit certain molecules. These are known as cell adhesion molecules. These cell adhesion molecules they tell the leukocytes that this is the spot where I need to leave the vessel and go in the extracellular space. And how do they go in the extracellular space? The process is known as diapedesis. What is diapedesis? They extend their cytoplasmic arms just like amoeba, and then after one arm they extend their another arm, and then they squeeze through the endothelial cells into the extracellular space. And then in the extracellular space also, how do they keep on following the chemical substances, and this is known as chemotaxis. Just like we have GPS, they have chemotaxis. Those chemicals they tell them where to go. And once they reach there, initially we have neutrophils, and then later on the macrophages they also come in, and we know macrophages, they are monocytes, they come in and then they move there. Macrophages they stay for a longer period of time, and macrophages they are important in both acute inflammation and chronic inflammation. So initially we have neutrophils, but later on the macrophages they join in. Okay, so this slide it summarizes everything we went over. It is in your Pearson e-text. If you want a revision, you can go over all the steps of this slide. I will not go over it again because we have already discussed these things. Okay, now let's see this clinical scenario of inflammation. What is pus? Pus is a mixture of dead or dying neutrophils, broken down tissue cells, and living and dead pathogens. So, at the site of infection or inflammation, a pus is formed, and this is what it contains: dying neutrophils, broken down tissue cells, and living and dead pathogens. And what is an abscess? If the inflammatory mechanism it fails to clear the area, the collagen fibers they surround that spot, and that pus it is sealed off. So now it is known as abscess. So just like let me draw here. Let me take this. So for example, these are those organisms and those cells, and the body it is unable to seal them or get rid of them. I mean, not to seal them. So what happens? Then the collagen fibers are put on around them, and now we have this closed thing. This is known as abscess, and the surgeon has to surgically open the wall of the abscess and drain it, because the antibodies they do not diffuse in the abscess. Then there is another term, granulomas. Some bacteria they survive inside the macrophages. So macrophages, when they engulf those bacteria, the macrophages are unable to kill those bacteria. So what happens then? A granuloma forms. Those infected macrophages they're surrounded by healthy macrophages, and then there is a fibrous capsule. Tuberculosis bacillus it has a habit of doing this, and the danger is that when the patient's immunity is low, when that person's immunity is low, these granulomas they can become active and cause problems. So we are doing innate immunity, and we have already discussed the phagocytes, the natural killer cells, and the inflammation. Now we have certain proteins which are involved in innate immunity. They are antimicrobial proteins. It means that they can kill the microbes. They are interferons and complement. So where do interferons come from? Viruses, when they cause infection, how do they do it? Certain parts of the viruses they enter the cell. Then they go into the nucleus. In the nucleus, they take over host machinery. So now that cell, instead of making its own proteins, it is making viral proteins, and it is like serving the virus instead of serving itself. So this cell, to warn other healthy cells, it secretes certain substances which are known as interferons. So these interferons, they do not help this cell which is making them. It is to warn other cells. So these interferons, they go to other cells, and then they cause that cell to make some substances which stop the infection of those cells. So now the virus cannot infect another healthy cell, or which because of the interferon, that cell, it has developed resistance. So these are interferons. And interferons, they can be alpha and beta, or they can be interferon gamma. So why have I written them separately? Because interferon alpha and beta, they are secreted by many cells, and they have many antiviral effects, and they also activate natural killer cells. We know when we went over natural killer cells, that natural killer cells, their target cells are the viral infected cells, and the cancer cells. And then the interferon gamma, it is secreted by the lymphocytes, and it has widespread immune mobilizing effects. It activates the macrophages. And we have interferons, they are being used against cancer treatment, especially in hepatitis C, which is due to hepatitis virus C, that can lead to cancer. So hepatitis C and genital warts, in also in genital warts, which is a viral infection. So interferons, they are used to treat viral infection, and also they can be helpful in certain cancer treatments. This brings us to our second type of antimicrobial proteins. They are known as complement proteins. There are about 20 complement proteins in the plasma, and they are activated by three pathways. There is a classical pathway, then there is a lectin pathway, then there is an alternate pathway. Classical pathway is through antibodies. These antibodies, they activate these complement proteins when they bind to the target cells. Then there is lectin pathway. Lectin, it is a substance which binds to the sugars in the microorganism. And so this lectin pathway, it also activates the complement system. Then there is an alternate pathway. Alternate pathway, when a cell, it binds to the microorganisms, the microbes, this alternate pathway, it is activated. All three of these pathways, they target C3. C3 is a complement protein. It is, when they target it, when it is activated, it splits into C3A and C3B. Now this C3B is an opsonin. Opsonin, remember we went over opsonization earlier in this topic, when I told you that certain proteins, they coat the top of the microorganism, especially those which are capsulated, and because the phagocytes, they cannot detect those microorganisms. So these opsonins, they, in a way, they mark them that this is your target cell and you need to eat it. So this process is known as opsonization. C3b, it is an opsonin. It sticks to the surface of the microorganism, and now the phagocyte, it becomes easy for the phagocyte to recognize it. And at one end, it sticks to the microorganism, and at the other end, the phagocyte is able to stick to this complement protein C3b. And then the other portion, C3a, it, along with another protein C5a, it is very important in chemotaxis. Chemotaxis, like we just discussed, that how do leukocytes know where the injury is and where to go. So these are certain chemicals and certain proteins, they guide the leukocytes. So this C5a and C3a together, they make very potent chemotactic agents, and then they also play role in other parts of the inflammation. Then there is these complement proteins, they make membrane attack complex. So membrane attack complex, these are the complement proteins which make them. And this membrane attack complex, what does it do? It drills a hole in the surface of the microorganism. So this plasma membrane lipid bilayer, it is microbes plasma membrane over here, and this membrane attack complex, it is drilling a hole in it, and when it drills a hole in it, because it is an aqueous environment, the water, it pours in it, and in this way the microbe, it is killed. So these are our complement proteins. Now this brings us to the last point of our second line of defense, which is fever. So fever, it is an abnormally high body temperature, and it is a systemic response to the invading microorganism. In our brain, we have a temperature regulating a part of our brain which regulates the temperature. So what happens when there is inflammation going on and a pathogen has attacked? The WBCs and the macrophages, after exposure to foreign substance, they secrete certain pyrogens, and these pyrogens, they go to the body's thermostat and they tell the thermostat something is wrong. So the body's thermostat, it increases the temperature, and this results in fever. Now fever has these effects. It speeds up the repair process, it increases the metabolic rate of the tissue cells, and then there is another effect. It causes the liver and the spleen to sequester iron and zinc. So liver and spleen, they just absorb the iron and zinc from the blood, so bacteria cannot use it. So fever, in this way, it helps the leukocytes to do their job. Now this table, it summarizes everything we went over. You can read it. It will be like a summary. It is in your Pearson text.